So, please can I introduce our third speaker for this morning. We've got Dr. Timofey Skvortsov, apologies if I've butchered that, from Queen's University Belfast, and his talk is entitled Bacteriophages as Living Antimicrobials.
So, thank you very much for coming to my talk. And I think to start with, I have to apologize slightly for the title of the talk because I'm using living and bacteriophages in the same sentence, and people who are working on phages might have a couple of questions to me about that. So, I will start by maybe introducing what phages are and why applying living might not be the best idea to them. So, phages are bacterial viruses. They look a little bit like a monoarthlander with a large head which contains the genetic material of a phage and the tail which contains all the apparatus, all the necessary attachment features that allow the phage to attach to the bacterial cell, introduce its phage DNA into it, which causes the initiation of biosynthesis of phage proteins, followed by assembly of new phages, and finally the untimely death of the bacterial cell, which releases the phages into the media, right? And these phages can go on to infecting new bacteria, replicating in them, and so on, and so on. So this essentially makes phages quite useful for us because they combine the following features. They are specific and efficient bacterial killers. They can shape the structures of microbial communities through killing of specific bacteria. They represent a reservoir of untapped genetic diversity. We still don't really understand the functions of many phage genes, and we keep exploring them. We keep finding new useful functionalities. And they are natural, ubiquitous, and harmless to humans. Again, harmless maybe is a very strong word here, but they're mostly harmless. And what's probably I should also say that phages can work against antimicrobial resistant bacteria. So all this makes phages quite useful antimicrobials. We can use phages directly as a form of phage therapy, just applying phage on their own. We can use phage enzymes, for example, the lytic enzymes that are produced by phage at the end of the life cycle, which kill bacteria, or we can use phages and their enzymes for dispersal of biofilms, right? And in today's talk, I'm going to give 3 examples of how we can use phages to treat bacterial infections both in humans and in the environment. So I'll start with talking a little bit about catheter-associated urinary tract infections. So it's quite a common type of infections. Urinary tract infections are probably one of the most common type of infections just in general. And catheter-associated urinary tract infections occur when pathogenic microorganisms colonize catheters, for example, after surgeries, long-term catheterization necessary to allow for the outflow of the urine. And there are many pathogens that can cause urinary tract-associated, catheter-associated urinary tract infections. So Escherichia coli and Klebsiella would be 2 big nasties in this area. But I want to talk specifically today about Proteus mirabilis. So Proteus mirabilis is a really interesting microorganism. It's a gram-negative bacterial species. It's present in low numbers in our intestines. But when a patient is catheterized for a long time, Proteus mirabilis typically replaces or other microorganisms and causes formation of biofilms. And these biofilms are quite special. So Proteus forms what's called crystalline biofilms. You can see here on the right the Foley catheter. It's the type of the catheter they used for urinary catheterization. And Proteus causes formation of biofilms that are crystalline. So essentially it blocks, this crystalline biofilm blocks the lumen of the catheter. They prevent the flow of urine, which causes back pressure, and that can lead to the ascending of the bacteria into the urinary tract and to kidneys and long-term infections. So this process happens in the following stages. First, bacteria attach to the surface of the catheter, start forming polysaccharide layer surrounding them. Then, to the action of urease, pH increases. This leads to formation of diffuse crystalline material, to precipitation, and finally formation of large struvite and apatite crystals that you saw on the previous slide, right? So what we decided to do, because Proteus mirabilis is a bacterial species and we work on phages, obviously we wanted to find a phage that can kill Proteus mirabilis. And that's pretty much how our lab started. Cormac, my first PhD student, isolated the phage that he named after himself, which is sometimes a good idea. And what's interesting about this phage can kill Proteus mirabilis. You can see here it forms plaques. Plaques are small pinhole zones of clearance of bacterial loam. So that's a bacterial loam of Proteus. But in addition to that, Proteus mirabilis phage forms halos. So these halos are typically a result of action of another enzyme that phages produce. And this enzyme degrades the polymeric matrix of the biofilms. And we decided to study that in further detail. We found the gene encoding this enzyme in the phage genome. We isolated it, we purified it. We compared its action in vitro to the phage itself. You can see here that, well, probably you cannot really see it too well here, but it remains active up to 1 nanogram per milliliter, which is quite good. And in comparison to phage itself, it doesn't really kill bacteria, but it degrades the biofilm. And we even tested that this enzyme in Galleria melanella model. We infected Galleria melanella larva with Proteus mirabilis. So we learned that Proteus mirabilis is absolutely deadly to larvae. Even 4 bacterial cells per larva can already kill a significant number of larvae. And we demonstrated that this purified spike provided partial protection to Galleria. So obviously it didn't completely protect Galleria from bacterial infection, but it managed to suppress its virulence in this model. So we got encouraged by these results and decided to see how we can use this phage PMC jar in combination, which produces dipolymers in combination with something else, because phages can be used in combination with other antibacterials. And we could have tested the effect of the phage in combination with antibiotics, but instead we decided to try something new, something interesting, and we tried cold plasma activated water. Again, I'm going to give a quick explanation of what it is, but later today my colleague Dr. Thomas Thompson will have a really good talk about cold plasma, so please stay tuned to that. So cold plasma activated water is the liquid of water in this case that contains reactive oxygen nitrogen species that form in the water upon the treatment of water with cold plasma discharge. So cold plasma discharge ionizes the water. And we conducted a very simple experiment. We allowed Proteus to grow in catheter pieces, form biofilm, and treated it with cold plasma activated water first and then phage, and vice versa, with phage first and followed by water. What we found that the order of treatment actually was really important. When we treated this biofilm or with the plasma activated water or phage on their own, or the combination of the phage and water with phage used first, that we saw some reduction, log reduction in bacterial numbers, but it wasn't that impressive. But when we changed the order of treatment, when we treated the biofilm first with the water followed by phage, the bacterial number reduction increased to 4 log. And as you can see here, we almost completely managed to eliminate the biofilm. So this order of treatment, plasma activated water followed by phage, really helped us to disperse biofilm. So again, we got quite encouraged by the results we were getting with cold plasma, so we decided why not to use cold plasma a little bit more. And my student Akash, collaboration with Professor Brandon Gilmer, lab conducted another type of experiment. We decided, why can't we just simply attach the phage to the surface of the catheter? So good idea, doesn't work on its own. We need somehow to fix phage to the surface of the catheter. And for that, we used cold plasma. We treated the surface of the catheter with cold plasma first, immersed these catheter pieces into phage-containing solution. Phage got attached, and what you can see here on the right, I think it's the best type of experiment to show. This is called the catheter bridge model. On the left, you see a piece of catheter that was just treated with plasma, and to which we attached phage. So Proteus mirabilis cannot migrate through this catheter bridge from one piece of agar, nutrient agar to another, while if we just immerse this catheter piece into phage, Proteus mirabilis can perfectly fine colonize this catheter piece and move on to the other segment of the agar plate. So again, this is something that we keep working on, and I think we getting quite interesting results here. But now I want to talk about another projects that we have. We working in addition to, 1 minute. Okay, so we work on as well acne, and for that we actually also work on microneedles as well, which allows us to deliver phages into the skin. Again, today we already heard about microneedles. This allows us to bypass the protective layers of microneedles and deliver phages into the skin pores, killing Cutibacterium acnes, causing acne. And in addition to that, one of my other students working on a different type of phage delivery based on the use of hyaluronic bicarbonate and hyaluronic acid combination. So we managed to demonstrate that this combination allows us to deliver phages under the surface of the skin and concentrate phage where it can act on Cutibacterium acne. And our final project is completely different. We are working also with collaboration with people from biological sciences on harmful algal bloom. We develop a phage that can suppress growth of cyanobacteria in the lake. And at the moment, we have a phage cocktail that we demonstrated can be used for that. So again, just 3 examples of how we can use phages to treat both environment and human-associated diseases. So thank you very much to my colleagues, to my funders, and obviously to my lab. Again, if you want to learn a little bit more about what we're doing, please visit our site. Thank you very much.
Brilliant. Thank you, Thomas.